Hi guys, welcome to TIMS channel. This is the second physiology lecture for the CNS module. In this lecture, we are going to be talking about the types of ANS receptors and describe the role of ANS when controlling everyday life situations. And these are the objectives. We may divide ANS receptors into cholinergic receptors or adrenergic receptors based on the type of neurotransmitters that bind to them. Cholinergic receptors are a type of receptors to which acetylcholine binds, while adrenergic receptors are a type of receptors to which norepinephrine or epinephrine binds. Actually, this is how we described neurons as cholinergic or adrenergic last lecture based on the type of neurotransmitter they release. The combination of neurotransmitter and receptor functions as a signal to cells, causing them to respond. Depending on the type of the cell, the response is excitatory or inhibitory. However, before going any further through the subtypes of the receptors, there are two important terms we need to know. The first one is agonist, and this refers to a substance that binds to specific receptors and activate them, while the other term is antagonist, or like the agonist. When antagonists bind to receptors, it prevents them from being activated. Okay, now adrenergic receptors. They are located in the plasma membranes of effectors innervated by sympathetic division, and the response of cells to epinephrine or norepinephrine is mediated through G proteins. Adrenergic receptors are of two types, alpha and beta. Alpha is subdivided into alpha 1 and alpha 2. Alpha 1 adrenergic receptors. As any adrenergic receptors, alpha 1 receptors are G-coupled protein receptors. They are found in the vascular smooth muscles of the skin, splanchnic regions, GIT, bladder sphincters, and radial muscle of the iris. Binding of norepinephrine or epinephrine to these receptors lead to the stimulation of the phospholipase C, IP3, and intracellular calcium. So, when calcium is released into cytosol, it mediates a cascade to result in excitatory response inside these cells. Alpha 2 receptors. Same as alpha 1, they are GPCR receptors. However, it has an inhibitory effect. This means this time binding of neurotransmitter to the receptor activate G inhibitory protein. These inhibit adenylate cyclase, which lead to decrease in cAMP, which is cyclic AMP inside the cytosol, leading to inhibitory effect, either by relaxation or dilation. Alpha 2 receptors are found in autoreceptors, platelets, fat cells, and heteroreceptors on the walls of the GIT tract. Beta receptors. Beta receptors are also subdivided into beta 1 and beta 2 like alpha receptors. Beta 1 receptors are found in the conductory system of the heart, SAN and AVN. When epinephrine or norepinephrine binds to P1 receptors on cell membranes of cardiac myotomes, they result in excitatory response, which is increased heart rate. Actually, their mechanism of action is that opposite for alpha 2 receptors. Beta 2 receptors are found in the vascular smooth muscles of skeletal muscle, bronchial smooth muscle, and walls of GIT and bladder. They have inhibitory effect, so relaxation is produced. Now, let's see the second type of receptors, cholinergic receptors. There are two types of cholinergic receptors, nicotinic or muscarinic. Before going through them, we need to know something. The classification of these receptors is based on laboratory finding that nicotine in tobacco can bind to some cholinergic receptors, while muscarine can bind to others. And here how they got their names. Let's start with the nicotinic receptors. They are of two types, N1 and N2. N1 are found in the neuromuscular junction, while N2 are located in cell bodies of both sympathetic and parasympathetic postganglionic neurons in autonomic ganglia. When acetylcholine binds to alpha subunits of these receptors, the receptor would open an ion channel for both sodium and potassium, leading to either an excitatory effect by increasing sodium concentration inside or inhibitory effect. Now the muscarinic receptors. They are located in the membranes of effector cells that respond to acetylcholine released from postganglionic neurons. From here, we can expect that they are most commonly respond or located to parasympathetic neurons, except for sweating glands. There are three types of muscarinic receptors, M1, M2, and M3 according to their location. M1 are found inside the CNS, central nervous system, M2 heart muscle, and inside the heart muscle they have inhibitory effect. M3 are located inside smooth muscle or glands. Here they have excitatory effect. For the heart, G inhibitory protein works for inhibition of adenyl cyclase, leading to opening of potassium channel, slowing the rate of depolarization. Please remember that the response of the heart for P1 receptors is excitatory, while for M2 is inhibitory. For smooth muscles and glands, they have the same mechanism found here. For the alpha 1 receptors. This table is made to make it easier for you to remember the differences of adrenergic and cholinergic receptors. Here for this table, we just need to know type of receptor plus its location. So we need the two first columns. Remember the functions of the ANS: sympathetic, fight, fright, and flight; parasympathetic, rest and digest. Again, note the function depends on receptor type in effector organ and neurotransmitters. This table shows you the effect of autonomic nervous system on organ systems. We are going to have two examples: heart and bladder. Try to read the rest of these organs, and if you found anything tricky, do not hesitate to ask. Let's start with the heart. Imagine you have a flight. So now the sympathetic nervous system had the over control. I need to increase my heart rate. So I need to use beta-1 receptors so that I can affect my heart rate. In the opposite way, when I'm relaxing, reading a book, my parasympathetic nervous system had the over control through M2 receptors. So we need to decrease the heart rate. Bladder. During a fight, of course we cannot go for the toilet. So we need to inhibit such an action. This occurs by sympathetic nervous system by two ways. The first thing, to relax bladder wall. Why to relax it? Just imagine when the bladder wall contracts, actually it pushes the urine out. So when it relaxes, it cannot push anything. So we need to relax it. Please remember which receptor is responsible for wall of the bladder relaxation. It was P2. Plus constriction of the sphincter. And here the responsible receptors is alpha 1. Alpha 1 was responsible for the sphincter to give excitatory effect by constriction. What if I'm eating in a restaurant, drinking juice? Now I have the ability to go for the toilet. So I need my gallbladder to contract and the sphincter to relax. In this case, acetylcholine is our neurotransmitter, and of course M3 receptors are the way to reach the cells. Here is the final part of our lecture today: Ans regulation. As anything in our body, Ans need to be regulated. Actually, Ans main function is to maintain homeostasis, and its regulation is through two divisions: higher centers in our brain and normal reflexes. Higher centers like hypothalamus, medulla, pons, these are there to control unconscious things controlled by ANS. For example, hypothalamus is responsible for controlling hunger, temperature. Respiration is controlled by ANS, which is regulated by pons and medulla. For ANS regulation for the cardiovascular system, we need the medulla. This table actually shows you CNS components in relation to their effects on the ANS. Please read it, and if you found anything tricky, just write in the comments. The reflexes. Inside the walls of our arteries, there are something known as baroreceptors or stretch receptors. Actually, they are there to detect any changes in our blood pressure. For example, sensory neurons transmit impulses from these receptors through glossopharyngeal and vagus nerves to the medulla oblongata. As we said, medulla is responsible for the cardiovascular system. Inside the medulla, interneurons are responsible for the integration of the information, and it transmitted to the motor nerves. Then, either a parasympathetic or a sympathetic reflex is going to control the action. For example, a sudden increase in blood pressure would increase a parasympathetic reflex. Remember, parasympathetic leads to decrease in heart rate, so increase in blood pressure requires reduction of heart rate, so that bringing blood pressure into normal value. Another thing is that what if my blood pressure decreased? The opposite way round. Sympathetic reflex will be initiated, of course. Again, the same reflex occurs. Stretch receptors feel a decrease in blood pressure. Sensory neurons transmit information to the medulla. Inside medulla, interneurons integrate the information. Parasympathetic nerves carry an impulse for the heart, causing it to increase the rate by increasing force of contraction, and thereby increasing blood pressure again. Here we reach the end of our lecture today, and thank you.